Hi everyone, I'm Elsa Mikkelsen, and today I'll be talking about using whole genome sequencing to reveal the evolutionary history of an enigmatic seabird family. Hybridization between species is widespread in nature. When closely related species interbreed, they may produce hybrid offspring. Often, these hybrids have lower fitness than their parents and are unable to produce offspring, maintaining boundaries between species. In many cases, however, hybrids are able to reproduce and can pass genetic material between their parental species through the process of introgression. This can allow the sharing of adaptive variation between species through the process of adaptive introgression. For example, the mimetic color forms of heliconius butterflies provide a striking example of adaptive introgression, with complex wing pattern traits passed between species through hybrid intermediates. In its most creative form, hybridization can even lead to the formation of new species through the process of hybrid speciation, in which a hybrid lineage becomes reproductively isolated from its parental species. Now, there are relatively few cases of hybrid speciation known in birds, but advances in the ease of genome sequencing means that it's easier than ever to test cases of hybrid speciation. Since the late 90s, there's been another proposed hybrid species of bird within the family Stercorariidae, which has remained unconfirmed in the absence of genomic data. In this project, we'll use whole genome sequencing to support or reject a hybrid origin for this species. The family Stercorariidae contains 7 species of piratic seabird distributed throughout the oceans of the world. They breed throughout the highest latitudes of the northern and southern hemispheres, but wander widely over the oceans during the non-breeding season. Traditionally, the stercorariids have been placed into 2 clean-cut phenotypic categories, the Jaegers and the skuas. Despite their distinct appearances, though, careful behavioral observations have shown that the Pomeranian Jaeger shares many traits with the skuas that are not shared with the Jaegers. In addition, genetic evidence from the mitochondrion suggested a particularly close affinity between the Pomeranian Jaeger and the great skua in particular. And based on this, it was proposed that the Pomeranian Jaeger could be a species of hybrid origin. To investigate this, we sequenced the whole genomes of all 7 species of Stercorariidae using shotgun resequencing of samples from the Royal Ontario Museum. We then mapped these sequences to the chromosome-level assembly of the razorbill, a member of the sister group to the stercorariids. We used this genome-wide dataset to accomplish 3 goals. Firstly, to resolve the species-level phylogeny of the Stercorariidae. Secondly, to determine whether a hybrid speciation scenario could be supported for either the Pomeranian Jaeger or the great skua. And thirdly, to assess evidence for introgression amongst the stercorariids. The first thing we did was to address the species-level phylogeny for the group, using genome-wide data in a multi-species coalescent framework. In this phylogeny, the 4 skua lineages formed a shallowly diverged monophyletic group, sister to the Pomeranian Jaeger. The 4 skuas are estimated to have diverged very recently, with the earliest divergence estimated to be as early as 90,000 years ago, remarkably young for bird species. So next we built a mitochondrial phylogeny, shown here in blue. In conflict with the species topology, the mitochondrial phylogeny places the great skua sister to the Pomeranian Jaeger, matching the contested topology from previous studies. With this mitonuclear discordance, there are 2 plausible causes. There could be incomplete lineage sorting, or there could be introgression. Incomplete lineage sorting happens when ancestral variation is retained past more than one speciation event, in which case it's possible for 2 non-sister lineages to inherit the same alleles. Now we can differentiate between incomplete lineage sorting and introgression based on the structure of the gene tree. Under incomplete lineage sorting, the discordance in the phylogeny is due to ancestral variation in the common ancestor, and so the discordant haplotypes of the Pomeranian Jaeger and the great skua would have to coalesce in their common ancestor before their species divergence date 1 million years ago. However, if introgression were the cause, then the haplotypes might converge more recently, with the minimum age determined by the timing of introgression rather than the timing of speciation. So to determine whether the divergence date of the haplotypes could be consistent with incomplete lineage sorting, we plotted the distribution of genetic distances of mitochondrion-sized blocks of the nuclear genome, standardized for mutation rate. 4 overlapping distributions are shown here in gray, showing the nuclear distances between the Pomeranian Jaeger and each of the 4 skua species. The black line shows the standardized distance between the Pomeranian Jaeger's mitochondrion and the southern hemisphere skua mitochondria, which falls within the expected distribution. In contrast, the red line shows the divergence between the Pomeranian Jaeger and the great skua mitochondria. Now, this is lower than more than 99% of the nuclear windows, and so we can infer that this pattern is unlikely to be produced by incomplete lineage sorting. Instead, the mitochondria appear to have introgressed from the Pomeranian Jaeger into the great skua.
This leads us to the question of what the functional impacts of this mitochondrial introgression might be on the great skua. As the cell's producer of ATP, the mitochondrion is critical for the physiological function of an animal and is a frequent target for climatic adaptation in birds. As it is possible that the great skua colonized northern Europe from the subantarctic relatively recently, introgression could have facilitated the transfer of a locally adapted mitochondrial haplotype to the great skua after its northern dispersal. Now, another feature of animal mitochondria are that they have a relatively high mutation rate, and this is predicted to lead to the accumulation of slightly deleterious mutations on the mitochondrion and the need for constant compensatory evolution. We find that the skua lineages have experienced a much lower effective population size than the Pomeranian Jaeger since their divergence, leading to a potentially lower efficacy of selection, and so introgression may have facilitated the purging of potentially higher mutation load on the mitochondrion by replacing the skua haplotype with the Pomeranian Jaeger haplotype. Now, alternatively, the introgression could have had no effect. However, there is some indication that there could have been a functional impact of this mitochondrial haplotype replacement. The Pomeranian and great skua haplotypes differ from the southern hemisphere skua haplotypes by 13 amino acid substitutions, and of these, 3 were predicted to have functional impact on their proteins using Provian, and one of these was predicted to have been subjected to positive selection using MEME. While physiological tests would be needed to confirm functional differences, this suggests that positive selection could have facilitated the introgression of this mitochondrial haplotype.
But next we wanted to ask whether this mitochondrial introgression was accompanied by nuclear introgression. So we used a variety of tests using the whole-genome dataset. These tests are primarily based on comparing 2 sister species to a third lineage. And if one of the sisters has introgressed with that third lineage, it may share more derived alleles, have gene trees with shorter branches, and have more frequent gene trees placing them together. And in fact, we found that there is a signal of likely introgression between great skua and the Pomeranian Jaeger. However, this signal is relatively weak and likely reflects only a very small proportion of the genome. The vast majority of the genome supports a monophyletic grouping of the skuas, sister to the Pomeranian Jaeger, and so we found no support for the proposed hybrid speciation scenario, instead just a small trickle of introgression. However, even with this relatively low level of gene flow, there could still be opportunity for adaptive introgression.
One strong candidate for a nuclear introgression in the Pomeranian Jaeger is the Mc1r gene, a protein-coding gene that's involved in controlling the expression of melanin pigments. Previous studies have shown that a plumage polymorphism involving light and dark morphs in the Pomeranian Jaeger is controlled by Mc1r, and that the dark morph allele of the Pomeranian Jaeger was more similar to the skuas than the pale morph was. And this led to the proposal that the pale morph allele could have introgressed into the Pomeranian Jaeger from one of the smaller Jaegers. Now with our whole genome dataset, we can zoom out into the flanking regions of this gene where there's much more phylogenetic signal to resolve the gene tree. And we found that the pale morph Pomeranian Jaeger falls out as sister to the skuas as expected, with no indication of introgression from the Jaegers. However, when we add that previously published dark morph haplotype of the Pomeranian Jaeger, it's nested within the skuas. And this pattern is consistent with introgression from the skuas introducing this dark morph polymorphism into the Pomeranian Jaeger. And so the small trickle of gene flow may have opened up opportunities for adaptive introgression of ecologically relevant traits.
Next, we looked more broadly across the Stercorariidae phylogeny to see if there was evidence of other introgression events. In addition to the introgression between the Pomeranian Jaeger and the great skua, we found evidence for introgression between the South Polar skua and the Subantarctic skua, and between the Subantarctic skua and the great skua, with these introgression events estimated to have affected about 11 and 14% of the genome, respectively. We also found evidence of more ancient gene flow across the basal node of the phylogeny, between the parasitic Jaeger and the ancestor of the skuas and Pomeranian Jaeger. Now, since these lineages are now nearly 10 million years diverged, this gene flow likely occurred far in the past when these lineages were more closely related. The parasitic Jaeger was approximately 3.5 million years diverged from the skua lineage when it split from the long-tailed Jaeger, and so we can infer that this introgression event likely occurred when these lineages were around 3.5 million years diverged, implying that reproductive isolation took a few million years to complete in this group. Zooming into the skuas, while the introgression amongst the southern hemisphere species was no surprise, the strong signal of shared ancestry between the great skua and the subantarctic skua was unexpected. These two species breed in opposite poles, and neither are known to cross the equator. And so it is quite difficult to imagine a high level of dispersal between these colonies on opposite sides of the globe. However, there are a couple of alternative scenarios that could explain this result without invoking ongoing gene flow between the subantarctic and great skuas. Instead, these scenarios invoke a second transequatorial colonization event. In one scenario, we start out with the ancestor of the skuas and the Pomeranian Jaeger in the northern hemisphere. A population then disperses to the southern hemisphere and establishes the skua lineage. This lineage then diversifies in the southern hemisphere. During this diversification, the ancestral subantarctic skua lineage then recolonizes the northern hemisphere, forming what will become the great skua. Now in the northern hemisphere, the great skua encounters and hybridizes with the Pomeranian Jaeger, capturing its mitochondrion. At this point, the great and the subantarctic skuas might be considered sister lineages. However, ongoing gene flow in the southern hemisphere obscures this signal, making the southern hemisphere skuas appear monophyletic. Now there is a second scenario, and that would be keeping the great skua in the northern hemisphere, and then having a second dispersal south, establishing what will become the subantarctic skua, and then that ongoing gene flow amongst the southern hemisphere skuas homogenizes their genome. The Stercorariidae has had a complex evolutionary history involving introgression throughout their evolution and multiple rounds of transequatorial dispersal between the poles. Thanks for listening, and I'd be happy to answer any questions in the comments section.